Okay, so we might start each. So this I did.
Okay. I'll just move this out of the way. I think I can.
So we moving your mom off the screen.
I think, yeah, that's better.
Okay.
Okay.
Okay, hi everybody, and welcome to what is our third seminar series this year. So this morning we have David MacHugh, who's a full professor of functional genomics here at the School of Agriculture and Food Science. So Dave grew up in Dublin, so he's from Dublin. He obtained his Biomedical degree in genetics from Trinity and in 1990. And he undertook his PhD research in animal genomics at TCD, which he completed in 1995. So as part of his postdoc, he was a Wellcome Trust research fellow in bioarchaeology, and he worked on livestock paleogenomics. And then in 1999, he took up his lectureship here in UCD and was appointed associate professor in genomics in 2009 and full professor in functional genomics in 2019. And he's also had appointments within the college. So he was a VP for Research, Innovation and Impact in CHAS, the College of Health and Agricultural Sciences, between 2018 and 2021. He is a very accomplished researcher. He's a Conway investigator, and he's a member of the One Health Centre. Broadly speaking, his research program focuses on functional genomics and on the development of novel disease biomarkers for bovine TB. Other research activities include livestock production genomics, population genomics of extent and modern livestock production, which is population. No. This is all very important to develop. And also collaborations with SRM Ryan Hill in relation to genomics and performance of traits in thoroughbred horses. He's widely published. He's over 160 peer-reviewed publications, and he's co-founder and scientific advisors to Zinto Labs and the IDGen Limited also, which is part of MSD and Health. So today, Dave is going to give us an overview of his research and a special welcome to his mom, Sheila, who's online.
Okay, thanks a lot, Adele. And just to, I actually graduated in 1988, so a bit older than you might be thinking. So I'm going to talk today, actually, the focus is going to be on work that really has a thread all the way back to my PhD work, which is population and now functional population genomics of livestock, particularly focusing on cattle. So I've actually included the word history in the title because with the high throughput, high resolution genomic data sets we have now for domestic animals, we can actually tease apart aspects of the history and the prehistory of domestic animals and their interactions with humans. So I think everyone is aware that Charles Darwin used domestic animals extensively in his works, particularly The Origin of Species, where the first part of the book was devoted to making the analogy between human-mediated artificial selection on domesticated animals in particular, but also plants, and how it helps to understand how evolution by natural selection could operate. So he had this extensively described, sorry, my pointer isn't working, it's just the screen, extensively described in The Origin of Species. And then 10 years later, he published another book where he went into these ideas in more detail, particularly as well, proposing the idea of domestication syndrome, where there's a suite of traits, biological traits that seem to be consistently present in domesticated animals, in particular, relating to behavior that seems to have knock-on effects on physical appearance, such as pigmentation. I'll talk a little bit about that later on. We should also note that Alfred Russel Wallace, in parallel with Darwin, in a malarial fever dream, came up with essentially the same idea at around the same time. Okay, so in terms of population genetics, which is really what work that I've been doing really since I finished college. This is a famous statement, nothing in biology makes sense except in the light of evolution, which was then riffed upon by Michael Lynch in Arizona State University, a very renowned population geneticist, quantitative geneticist, and genome biologist. Nothing in evolution makes sense except in the light of population genetics. And really, population genetics provides the mathematical framework for understanding how evolutionary change can happen. So apologies for quick revision, but just to remind you, we're dealing with the DNA sequence information in animal genomes, and we can use humans to represent this because, for example, the livestock species that I'm going to talk about essentially have the same size genome as the other mammal, the human. And although the chromosome numbers differ, the basic principles apply. So the DNA sequence information is present, the vast bulk of it in the nuclear genome, which in diploid cells consists of the haploid genome that was transmitted from the male parent, and then the haploid genome transmitted from the female parent. We also have the mitochondrial genome, which is exclusively maternally inherited, and I'll talk a little bit about that in relation to cattle later. And then also, sorry, the pointer's not working. And then also the Y chromosome, which is exclusively transmitted from the male parent to his male offspring, and has the gene for the primary trigger for male sexual development. Okay, so our work really goes back more than 35 years. In the late 1980s, Paddy Cunningham, who's an animal breeder and quantitative geneticist who worked in Teagasc and then became a professor in Trinity, and Paul Sharp, who at the time was working extensively on the evolution of the HIV virus and really became very well known for that work and other work relating to viral evolution. But they basically were inspired by work in humans that appeared in 1987, where mitochondrial DNA variation was used to reconstruct human evolution and supported or seemed to support the out of Africa hypothesis for recent human origins within the last 100,000 years. And this created a lot of media interest, for example, for this Newsweek cover from early the following year. But anyway, Paddy and Paul realized that they could use the same techniques to study the evolution, the history, and in particular, the domestication of cattle. So in terms of the situation before cattle were domesticated, so this is just after the end of the last glaciation, about 10,000 years before present. Based on zooarchaeological evidence, this is the distribution of the aurochs, Bos primigenius, which is the wild ancestor of domestic cattle. So there were three subspecies, although this picture is changing as we now have much more paleogenomic data and other evidence. But Bos primigenius primigenius, Bos primigenius nommaticus, and Bos primigenius autonomous. And essentially the questions were, at the time, which populations contributed to modern domestic cattle diversity? Is there evidence from cattle genetic variation as to where domestication may have taken place? And other questions. So we started this, Ronan Loftus started first in 1989. I started the following year. And then Dan Bradley started as a postdoc after he'd completed a PhD in medical genetics. So this is some pictures from the field work that we did around that time. I'll come back to these cattle later. These are Curry cattle. That's Lake Chad in the background where Ronan was sampling in 1990. That's me wrestling a crocodile in the Nile. And then that's Dan collecting blood samples from cattle in West Africa in 1992. So this work, we really, I suppose in many respects, we were the first to do this sort of work in cattle. So we were lucky that we were then able to get good scientific publications from this work. Even though, as I'll come back to later, the data generation at the time was incredibly time-consuming, cumbersome, and really quite expensive compared to what's possible now. But anyway, we published a range of papers, I suppose, culminating in the first 20 years with the publication, ultimately, of the first full genome sequence from the aurochs, which we published 10 years ago. And actually this work, I'm not going to talk about it today, but this work did provide good evidence that supported these ideas about domestication syndrome and the relationship between behavior and particular neural crest cells that contribute to neurobiology ultimately, and also are involved in other physiological and anatomical processes that seem to support this idea of a domestication syndrome. Anyway, the culmination of that work, I suppose, is summarized in these diagrams. Now, Dan's group went on and sequenced multiple aurochs genomes from across Eurasia, and this paper was published last year in Nature. But I suppose a synopsis of it is that the aurochs distribution showed that there were really four major groups based on paleogenomes and modern genomic data: a kind of West Eurasian group, a North African group that actually has its roots in the Fertile Crescent, and then a more distinct group which gave rise to Bos indicus cattle, zebu cattle, and almost certainly domesticated independently in the Indus Valley, maybe 2,000 years after the first cattle were domesticated in the Fertile Crescent about 10,000 years ago, which then gave rise to European cattle, Bos taurus, humpless cattle, and humpless cattle elsewhere in the world. But recently, there is now evidence that there was a distinct strain of aurochs in East Asia, which is now taxonomically defined as Bos primigenius sinensis, that may also have contributed ancestry to Chinese cattle. But the major split is Bos indicus and Bos taurus, which we were able to date that they shared a most recent common ancestor about just over 300,000 years ago, which is actually comparable, even though the time is different, to the split between Neanderthals and modern humans. And the genome differences between Bos indicus and Bos taurus are similar to the differences between Neanderthals and modern humans, even though Neanderthals and modern humans diverged maybe 700, 800,000 years ago. It's a consequence of the generation interval, because cattle have a shorter generation interval. So anyway, sorry, just this diagram here then just summarizes the migration of cattle out of these domestication centers, starting about 9,000 years or so from the Middle East and 7,000 to 8,000 years from Indus Valley. So one of the areas that we focused on was Africa, because Africa is really a natural laboratory for hybridization between Bos taurus and Bos indicus cattle. And this diagram is from a paper published 25 years ago now by Olivier Hanot and his group at ILRI. It's 2002, and it using many genetic markers, well, relative for the time, a significant number of genetic markers genotyped in many cattle populations. They were able to reconstruct a map of genetic ancestry where it was clear that the bulk of the Bos indicus or the zebu ancestry was in East Africa. Now, we also, a couple of years previously, did similar work, but we also looked at the mitochondrial DNA, the nuclear genome, or the autosomal genome, and the Y chromosome. And we found that African cattle retain taurine mitochondrial types. They have mixed ancestry across the continent, which essentially corresponds to what Olivier's group found a couple of years later. And then the Y chromosome, the zebu Y chromosome, has introgressed substantially into Africa. And we presume to this as a consequence of male-mediated gene flow, particularly perhaps through the trade routes in the Indian Ocean. Maybe male animals were brought, and obviously a single male can potentially have hundreds of offspring. So this seemed to be the most plausible explanation, although more recent work makes this slightly nuanced picture. So anyway, over the last 6 or 7 years, we've been dramatically, I suppose, extending this work, taking advantage of the new technologies that we now have at our disposal. And in particular, James, Ishmael, and Gillian. James was a PhD student in UCD who now works in Ludwig Maximilian University with Laurent Franz. And Dan, and also a gene editing company, where the Chief Scientific Officer is Tad Sonstergaard. We've put together a project where we're taking advantage of the thousands of samples that we accumulated starting in the early 1990s in Trinity, in freezers in Trinity, and essentially sequencing them, generating whole genome sequence data from them. And this has been possible because since the human genome sequence was published, almost complete human genome sequence in 2003, there's been a dramatic revolution really in the technology that's available for accessing genomic information. So when I did my PhD, these were the sorts of technologies that I had to use, and we didn't really have semi-automated DNA sequencers at that stage. But now, really, for the last not quite 20 years, but data generation has become remarkably easy and cheap and rapid and high throughput. And the bottleneck is now not the data generation, it's the data analysis. And really this has been our focus, I suppose, particularly during the last 10 to 15 years. So just to illustrate this, around the time just after the almost complete human genome sequence was published, the cost, if you wanted to get your genome sequenced from scratch, would have been around that time maybe 100 million euros or dollars. Whereas now, 20 years later, the cost for a mammalian whole genome sequence is about 400 euros to generate the data, and probably will be half that next year with new technological developments that have come on stream in the last year or so. So this has led to a situation where population genetics has become population genomics and really a fully fledged data science. So for example, in my PhD, I had just under 30,000 data points essentially based on microsatellite genetic marker genotypes in about 800, 750 cattle. James, who finished his PhD last year, with a similar type of PhD really, he had 23 billion data points, 23 billion single nucleotide polymorphism genotypes. And these are the simplest form of genetic variation, the single base pair substitutions, single letter changes in DNA that are particularly easy to determine using whole genome sequencing. So just in terms of data volume, I had an ice cube, whereas James kind of more or less had an iceberg. Now, John O'Grady, who's just finished his PhD and will be doing his Viva this week, we worked out how many data points he had for his thesis, and he has 350 billion, so more than 10 times more than James, and about whatever more than 10 million or 100 million times more than me, or certainly millions. So anyway, this is part of the reason why James was on the Genomics Data Science Centre for Research Training, funded by SFI, and also now funded by Research Ireland. Genomics has become a data science, and so really the challenges are managing, analysing and interpreting the data. But what we're doing with this new project is using whole genome sequence data from about 5,000 animals. We have a global reference panel, most of which comes from the samples that we collected during the 1990s, and then Dan's group continued to collect for the following 20 years or so. Now we sequenced a proportion of these at high coverage, at high depth, which means we are sequencing with high fidelity and accuracy. But because of the way genomes evolve, and because of the way genetic variation is transmitted from parents to offspring, and this remixing that happens during gametogenesis, it's possible to reconstruct whole genome sequence data from relatively low accuracy sequencing data. And this process is called imputation. So we're using a mixture of imputed and whole genome sequence data, but we have about just under 12 million genetic markers for this study. And in particular, I'll focus on Africa at the beginning. In particular, it's allowed us to really put African cattle diversity under the microscope. So just to, I suppose, the analogy for genome imputation is really in the same way that a 4K or an Ultra HD flat screen TV can interpolate and use intelligent algorithms to fill in the gaps to make a lower resolution picture or video appear more high resolution. It's essentially the same idea. We can impute from lower density data, usually just single nucleotide polymorphism SNP data, all the way up to whole genome sequence data. And what we're doing with this is, I suppose if any of you have used 23andMe or similar genetic ancestry services, essentially we're doing 23andMe for cattle. And although we actually have about 10 times as many SNPs as 23andMe use for their service. So in terms of how we can visualize the data, in population genetics and genomics, one of the key tools is using principal component analysis to look for the major trends in the data. And this is work that Ishmael has done in his PhD and is currently under review. And we can see the three major axes of genetic variation in cattle, in African cattle. So we have European taurine up here, we have the Bos indicus cattle here, and then we have African taurine cattle here. And then the various admixed mixed populations that are the result of crossbreeding are in between, and the position on this two-dimensional surface corresponds to their various ancestries. And the bulk of the variation, for anyone who's familiar with principal component analysis, is determined, it's based on the first principal component, which accounts for almost 80% of the variation out of the first 10, showing the major difference between Bos indicus and Bos taurus cattle. Now, this recapitulates what we did back in the 90s, just using much smaller numbers of markers and just looking at it at a population level. These are individual animals, but essentially we're recapitulating what we saw with much smaller numbers of markers. Now, one thing that we've been able to do in collaboration with Michael Salter Townsend and Maths and Statistics in UCD is to start really dissecting this variation across the genome. So generally, you can look at an individual animal level, you can look at their ancestries in a plot like this, is called a structure plot. And each vertical bar here is an individual animal. And it shows the three major components of ancestry for a series of different cattle populations, European, African taurine, African Bos indicus. And essentially it's showing the ancestral contributions of the three major lineages to these populations. But with these really high density data sets and using initially lower density data, and Gillian MacHugh was able to develop methods with Mike's help to actually look at genomic ancestry at a subchromosomal level, so literally across the genome. And then start to look in particular regions that are retained in hybrid populations that may be a consequence of evolutionary processes such as disease, climatic adaptation, etc. So we were also using these kind of really high resolution subchromosomal data with almost 12 million SNPs. We can actually reconstruct the history of cattle in Africa in terms of their admixture, which is shown here on this heat map, which the taurine is yellow at its extreme here and dark blue or purple at this extreme. But also we can use this information, and based on how genomes get remixed as they're transmitted from parents to offspring, we can actually work out how many generations ago these mixing events started to occur. So we can convert it then into years before present. And it does seem to correlate reasonably well with what we know about the start of extensive trade in the Indian Ocean between Asia and Arabia and East Africa. So that the bulk of the Zebu ancestry is here, and the admixture started more than 1,000 years ago. And really, it really accelerated with the Muslim migrations into East Africa that started in the 8th century. Okay, now just to focus on one particular illustration of how we can use these data to uncover new information, in this case about potential trait relating to disease resistance. Going back to the cattle that Ronan was sampling in 1990 in Lake Chad. So this is a photograph of Lake Chad. Lake Chad is located here in Central Africa, North Central Africa. And the lake has been undergoing desertification for decades. And to illustrate this, this is the extent of the lake in 1984 from a satellite photo. And then about 7 years later, where it's reduced by more than 50% in size. So these cattle, now Lake Chad used to be much more extensive, and it was part of a much larger system of lakes and rivers in North and North Central Africa. But there's a particular, sorry, I missed it. Yeah, these cattle, the cattle, have adapted to living in the environs of Lake Chad. It's even proposed that their bulbous horns actually evolved or were selected, well, evolution via human-mediated artificial selection, as flotation devices because they spend a lot of their time in the water. And we can see the effects of desertification and the erosion of the Curry cattle genome over the course of 30 years from our material sampled originally in 1990, and then more recent population samples taken just 5 years ago, sampled 5 years ago. And this is what we call chromosome painting. So all 29 of the cattle autosomes are shown, and there's 2 of each because there's one male-derived one and one female-derived one. And then the color coding corresponds to the ancestry within each of those chromosomes. And you can see quite clearly that the Indocene ancestry is starting to dominate in Curry cattle that were sampled 5 years ago. And this is a consequence of the fact that the environment is changing, but also because the local pastoralists and farmers tend to prefer the Bos Indicus cattle for other reasons. And one particular analysis we did was to look to see the retention of taurine gene variants across the genome. And one that was particularly apparent in chromosome 5, and that's this peak here. So this shows a much higher taurine ancestry than expected based on the rest of the genome. We can also do statistical analysis for detecting selection at a genomic level as well, that illustrate that there's reduced variation at this gene in the Curry cattle. And it seems that a particular gene variant of the LRP1 gene, which encodes the LDL receptor-related protein 1, has been retained. And we would hypothesize, because it's been shown recently, that this is a host entry factor for Rift Valley fever virus, which is endemic in the area. And it seems, although we thought the smoking gun would be really easy to find, because usually with this sort of receptor-based mutation for resistance or resilience to viral infections, it tends to be easy to find because the receptor its amino acid sequence is normally changed. But we think this may be more subtle, and it may be just that there's an activity-based mutation that may be causing it to be reduced in expression in whatever cells this particular virus first encounters. But we're working on this to try and identify the underlying mutation at the moment. So that's an example of how this sort of genome-wide high-resolution data can really reveal things that just wouldn't be possible using older technologies. And we can do this because we're literally scanning the whole genome. Other work that we've done that I'm just going to touch upon, this was published several years ago, and this was work where we found clear evidence for mitonuclear co-adaptation in African hybrid cattle populations, so that the retention of the taurine mitochondrial type in Africa, although it was almost certainly influenced by the imbalanced sex ratios in terms of mating between males and females and the importation of Zebu Bos Indicus, bulls into Africa, almost certainly. There does seem to be evidence that the mitochondrial genome and the African mitochondrial genome is better adapted to the African environment. And this work was published 5 years ago, and it also, or sorry, 3 years ago, it also, James won for his infographic, he won a prize, and we got a kind of journal cover produced by a professional artist in the Conway scientific image competition, which sort of illustrates this point about mitonuclear co-adaptation. So I'm not going to talk about that. I'll talk about other work that James did in Criollo cattle as part of his PhD, where we were able to reconstruct the evolutionary history of Criollo cattle, which descend largely from Spanish cattle that were initially brought right at the beginning by Christopher Columbus when he landed at Hispaniola in 1492. But then later importations from Iberia, starting 500 years or more ago, and then Northern European cattle into North America, starting about 400 years ago. Now, we know that when we look at the genomes of these cattle, there is some African ancestry as well, which may have been something to do with the slave trade, and maybe cattle were brought as well. And then in more recent years, 150 years ago, Bos Indicus cattle were brought from India to upgrade cattle in South America to make them essentially better adapted to the environment. But particularly focusing on Criollo cattle in Colombia and Venezuela, combining it then with larger data sets that we had and other people had made publicly available, we were able to reconstruct the genetic ancestry in Criollo cattle. And I won't try and zoom in, maybe, I know you can't do it by touch, but in the principal components analysis, we found that they still shared a remarkable proportion of their ancestry that's derived from Spain, even after 500 years. Again, these are the sorts of structure plots. We can examine this ancestry in more detail. We're also able to use these chromosomal painting diagrams, and we can see the admixture from African taurine, from Asian Indocene and European taurine, all jumbled up over the last 400 or 500 years or so, and 150 years in the case of Bos Indicus. And you can see this is a larger chunk as a consequence of that. Now, again, we were able to use these data to detect recent evolutionary change. So we were able to detect a selection signature as a consequence of a previously described set of mutations that are present in these South American Criollo cattle that allows them to tolerate the really humid, hot conditions in South America. And this is called the slick trait, and it's a consequence of multiple mutations in the prolactin receptor gene that affects the structure of their skin and the structure of their coat, and has other physiological effects as well. So this actually has been particularly of interest to Acelagen, the gene editing company. Now, I should say we didn't discover these mutations. We discovered the selection signature in the animals that shows that this has evolved relatively recently. Okay, so I'm just going to finish off just by talking a little bit about other work, very quickly, other work that we're doing. So Gillian, as part of her PhD, also did functional genomics analyses of cattle that are trypanosome tolerant and trypanosome susceptible. I didn't really talk about trypanosomiasis, but it's a major disease challenge in sub-Saharan Africa for cattle, also for humans. But there are cattle in West Africa that have evolved a tolerance of infection with these trypanosomes. It doesn't mean that they clear the infection. They can tolerate the infection and essentially be as productive as non-infected cattle. Bos Indicus cattle, on the other hand, are trypanosusceptible, as are European cattle. But using functional genomics and using material that we actually worked on 20 years ago, that Kieran had a major role in, and Emmeline Hill as a postdoc then as well, we took these samples and then used newer technology to study them in more detail, and this was published earlier this year. And the idea now is, well, we've done it, but it's under review, integrate these data with our population genomics data to help us identify genes and genetic variation that contributes to this trypanosome tolerance trait in the West African taurine cattle. Another example of work, and this relates to another livestock species I've just been talking about, cattle. This is work that a really excellent PhD student on the CAS UCD collaborative PhD program did and published earlier this year, using a whole suite of functional genomics technologies, including single cell based approaches, to study how sheep got these fat tails. So there are certain breeds and populations of sheep that have these fat tails as a fat store, maybe as a consequence of natural evolution, pre-adaptation to particular environments, arid environments in Central Asia, and then accentuated by human-mediated breeding, human-mediated selection. Other work that we do that really provides a lot of our funding relates to TB, and this is work, relatively new project that John O'Grady has contributed to, and Adnan Khan, a postdoc who started with us last year, and also Kieran and Steve Gordon and Eamon Gormley in the vet school. And one of our outputs is published Task 5, which is basically taking a whole series of functional genomics data sets and other types of data from cattle, confirmed reactor infected cattle infected with Mycobacterium bovis, the pathogen that causes TB, bovine TB, and control cattle, and integrating these data sets together to try to reconstruct or to provide new information about the genetic architecture of susceptibility traits. Now, I won't go into the details, but essentially by integrating these data together, we can identify genetic variation and the genes and the genomic regulatory elements that regulate the activity of genes that contribute to susceptibility to M. bovis infection in cattle. And this was published earlier this year as well. And the other work then, just to briefly mention the work that I'm involved with Emmeline and Lisa. So it's really Emmeline who leads all the genomics, equine genomics work, but I've been lucky enough to be involved with it. And this is just an example from several years ago where, using the newer, really high resolution genomic data, we were able to fully confirm the myostatin gene, speed gene effect on chromosome 18, but also start to tease out the other genetic factors that contribute to race distance aptitude in thoroughbred horses. And at the moment, a lot of the work that Emmy's doing and that I'm involved with is now focused on inbreeding and the effects of the genomic effects of inbreeding in thoroughbred horses. Okay, so I'll finish there. Thanks a lot. There's obviously too many people here to read out all their names, but these are, I think, hopefully I haven't missed anyone, but everyone that's contributed to the work that I described, and also our funding and other support. Okay, thank you.
Thanks a million. That's really, amazing presentation, even the history piece. I'm just going to ask one really quick question. So if you look at, like, say, modern breeding techniques, and as we kind of continue to select for, things like production and the environment and rates and all that stuff, are there any kind of dangers? And I'm thinking about the kind of the fine bred animals in terms of disease or health.
Breeding is always a double-edged sword. So, for example, with intensive breeding, starting with the introduction of AI in the 1950s, in America at least, and also increasingly sophisticated tools for the quantitative genetic aspects of breeding. In the 70s and the 80s, there was the emergence, particularly of single gene defects related to the immune system and fertility, and then more complex phenotypic consequences that Pat will be familiar with regards to fertility. But with these new genomic technologies, breeding of livestock is now managed at a genomic level, essentially. So it's actually much more straightforward to manage inbreeding, but also to manage potential downsides with regards to deleterious genes or genetic variants that emerge because of inbreeding.
Yes,
so it is actually more straightforward than sort of paradoxically, because you still have to crunch loads of data.
Yeah. Okay, good. Is there any question from David?
I was going to salute. Where's the active zoologist? Yeah, from Mongolia.
Oh, right. Yeah. So there's a whole range of bovid species, and we're most familiar with like cattle as a domesticate, but yak were domesticated probably about 6,000 years ago in the foothills around the Himalayas. There's the banteng, which was domesticated in Southeast Asia. There's the gael, the mython. Another example is water buffalo, swamp buffalo. But we're most familiar with cattle. But the yak were obviously adapted to the Himalayas, and then they've admixed with cattle. So many of ostensibly yak populations around the Himalayas are actually admixed with cattle. And one thing that we do see, well, I didn't see it, but it's published, the genes that adapt yak to high altitude in these mixed populations, they're retained. So the cattle that have evolved to live in high altitudes have significant yak ancestry, particularly around the genes that allow them, to manage with low oxygen, for example, at high altitude. But they were domesticated about 5,000 years ago.
Okay, so long after.
Yeah, 5,000 or 6,000 years ago, but presumably inspired by domestication of cattle.
Karen,
I was just going to ask you about like some of the susceptibility loci that you've identified. Like, do you see a situation where it might be permissible to use CRISPR in Europe at some stage in order to make use of those kind of?
Yeah, I mean, first of all, gene editing is completely harmless from the point of view of human consumption of gene edited plants or animals. I mean, plants, there's no gene edited animals in the food chain, but gene edited plants are now increasingly being used. And they're extensively tested. They are tested in the same way that conventional plant-based food products are. Unlike many of the plant-based food products that we consume, they weren't produced by, mutagenic breeding, radiation breeding, that basically is a scattershot approach to generating mutation. But anyway, when it comes to animals, it's different. So I don't think in Europe we will see gene-edited animals in the food chain for at least 20 or 30 years. Completely contrary to the scientific evidence. And so it's ironic because the same people who, use the overwhelming evidence for human-mediated climate change will then completely ignore the overwhelming evidence for, the safety of gene editing or genetic engineering. So and unfortunately, they have too much influence in Europe. So
disease-resistant livestock will probably emerge in like America and other areas.
Yeah, they're starting to already. But even in America, even in South America, there have been problems, and there is increasing resistance because the global movement to prevent, agricultural biotechnology is influencing, other countries, not just Europe. It's increasingly. I mean, in Africa seems to have. I mean, the overwhelming benefits of these technologies in Africa is sort of countering the misinformation, the disinformation from the people who are opposed to this. So it is starting to happen. But in Europe will be the last place, I think, even though a lot of the technology was originally developed in Europe.
so will it be a case that we might not be involved in the development of it, but like we are with GMO, we're okay with importing it?
Yeah, well, I mean, like when genetic engineering first came into public consciousness in the late 90s, in the Irish supermarkets, there were tins of tomatoes that were proudly labeled as genetically modified, that had actually come from America, where the flavor saver tomato had been a disaster, and they just turned them all into tin tomatoes. And then they dumped it on Europe, and there was never any issue with it. It was only then that it became a problem because of misinformation and disinformation. But I think we're still eating GMO. I mean, I know that like the soy, whatever all the soy that goes into the food chain that ultimately ends up in Europe. I'm not talking about feeding animals, I'm talking about human. A lot of that is coming from genetically engineered soy plants, and so what?
Yeah, and you think we'll be okay then with importing genes from other countries, say, as they get more developed, like here in the States?
Oh, so for example, if in Africa, a lot of the vegetables that are grown in Kenya, for example, if they start to use gene editing technology to enhance.
Well, even in animals, so if we're importing.
Yeah, I don't think. No, I don't. They won't allow gene edited animals into Europe. Probably for at least 20 years.
David, what about what we've seen with gene edited bull in Australia? I mean, would we be able to detect that it was gene edited?
Yeah, you could detect the edit, but a lot of the time the edit will be a natural sequence variant that then, but it may be embedded in a particular sequence background that you could detect that would differentiate it from the natural sequence variant. But yeah, I mean, the gene editing companies, for the most part, have tried to focus on natural sequence variants because that's, you know, easier to overcome resistance. So the SLIC, and some of the, sorry,
The bulging.
the Paul-gene, yeah. So the Paul, yeah, but again, they haven't really worked because there's just so much resistance, and not just in Europe, but you know, particularly for animals, not so much for plants.
So any last minute questions for Duke?
Yeah.
Yes.
Yeah, thanks, David, for a very interesting presentation. You mentioned about how genomics is evolving very much into almost like a data science.
Yeah.
Could you maybe say a little bit about kind of what major developments you see down the line there? I mean, we hear a lot about things like machine learning, for example. Is that what you're referring to there? That's
part of it. So part of John O'Grady's PhD work was using machine learning to identify biomarkers of infection from functional genomics data, the same data set that I showed you related to the TB work we've done. But also, I suppose, there's the area of what's become known as synthetic biology, where because the genome is essentially it's just an information, it's a repository of information, and the technologies for it's not just about sequencing or examining the activity of genes. It's increasingly about rewriting genomes, and gene editing, I think, is just the start of that. So, for example, machine learning is impacting on in silico, completely predicting what gene edits you can use that would enhance a particular trait. And ultimately, I think, you know, you'll have in silico models of animals at a genomic level, and you know, you can do a lot of this in silico, and that's obviously going to be very much driven by data science. But that'll be long after I'm finished.
right.
Okay. Well, there's no other questions. Just want to say thanks,
Thanks,
David.
David.